can be very different. They can be, for example, different types of carnivores like lions or crocodiles, or they could also be linked to human activity such as butchery. So these studies are quite frequent in archaeology and paleontology because they can give us a lot of information about the different kinds of animals that lived around a site or about the different kinds of human activities that were done on each site. However, there's never been a study on the tooth marks for carnivores and notably for crocodilians. So this study is to see basically whether tooth marks can give us any new information and whether it's worth integrating them into archaeological studies. So the main questions we ask are there any correlations between the morphologies of tooth marks and the tooth apices that created them, and what can this teach us about the individuals that produced these marks? For this study, 4 extant species were studied. Alligator mississippiensis, Crocodylus niloticus, Mesostops cataphractus, and Osteolemus tetraspis. There was also one extinct species, Goniopholis, who comes from the Angeac-Charente site in the Charente, which is central France, and is an early Cretaceous site. The specimens that I used for my study, the extant bite marks were from 2 studies by DRUMHELLER and Brochu in 2014 and 2016. They kindly sent me the molds that they used for these studies, which I then scanned. The thing to note about these bite marks is that they were taken using crocodiles in captivity. Therefore, there are a couple of biases. Most notably here, you can see that certain individuals were restrained during the feeding process, and this can create a few biases that we talk about later. The teeth that were molded are from the MNHN's collections, and then all of the Goniopholis apices and bite marks are from the Angoulême Museum. So the first step of studying the specimens consisted of deciding which teeth we were going to mold. So we determined some reference teeth, what we call reference teeth. These are all of the most prominent teeth in the crocodile's jaws. So if a crocodile closes its mouth, some teeth will protrude. They're bigger than the others. And because they're bigger, they're going to be the first ones to come into contact with the prey, and therefore the most likely ones to create marks. So all of these teeth were molded, and then we did one individual per species for all the other teeth, just to compare. I then described and measured all of the teeth to determine whether there were different morphotypes for different species, whether what the wear state was for each tooth, and to get general size data. The next step was to mold each of the specimens. So we did the teeth and we did the bite marks on the bones using high-resolution silicone. These molds were then scanned using a confocal microscope. In total, we have 155 apex scans and 260 mark scans. So this is what a scan using a confocal microscope looks like. As you can see, the data can't be used in this state. It has to be treated first because it has all these holes and outliers, which would be problematic. So to treat these scans, first of all, you need to mirror them because this is a mold and not a mark. Then the outliers need to be removed and the non-measured points filled in. And then finally, the profiles are placed all along the bite mark to get a general idea of what's going on inside. For the apexes, the process is exactly the same. The only difference is that there are only 4 profiles taken and not 10, because here what's interesting is the orientation of the teeth. So we have 4 orientations. So the next step, once we have all of our data, is to perform the statistical analyses. We can't do a typical morphometric study on this type of data because the traces don't have homologous points that you can refer to. So we decided we'd do an elliptic Fourier analysis, which is basically when you add ellipses into a shape until you get the whole shape. So for this study, we did a test and we can see that 6 ellipses were sufficient to explain 98% of our data, which means that all of our PCAs include 24 coefficients. So the first result that we got was on the measurements that I did directly on the teeth. I put it into Excel, and we can see that 2 morphotypes are defined. Let me see if I can, there we go. At the back here in gray, we have the back teeth. and here we have the front teeth in orange and blue. So this was the case for all crocodile species. They have two different kinds of shaped teeth, basically. We wanted to see if the signal was also visible on a microscopic level, and it is. Here on the left-hand side of the graph, we have all the back teeth grouped together, and on the right-hand side, all of the front teeth are grouped together. So this, the morphotypes are conserved microscopically. Next, we did a test on the BSMs using to test different species. These three species have very similar ecological niches. So it's understandable that they would all have very, very similar bite marks. However, on PC1 here we have depth. We can see that the alligator in black produces deeper marks, whereas the Goniopholis in blue produces the shallowest marks. The next test we did was on size. So in red here we have the small individuals, and they're all grouped together. There's not that much variability, whereas the black ones in which are the big individuals have much more variability. This time depth is on the PC2 axis, and we can see that the small individuals make shallower marks, whereas the larger individuals make deep marks. This makes sense because the larger the animal, the more power it has behind its bite. If you remember at the beginning, I said there were some biases linked to the fact that certain animals were restrained during feeding. So this is what we tested here. Once again, depth is on PC2 here. So the non-restrained individuals up here create shallower marks, whereas the restrained individuals create deeper marks. This is probably due to stress. If an animal is being restrained, it's more stressed and therefore will bite harder and create deeper marks. Now, what's interesting here is that all of the restrained individuals are actually smaller than the non-restrained individuals. This means that despite what we saw in the previous graph, animals that are smaller can still create deeper bite marks if they are more stressed than larger individuals. The impact of stress on bite mark creation has already been studied in other species. For example, in this paper by COURTENAY et al., they tested wolves to see, and they saw that basically in captivity, wolves create deeper marks than they do in the wild due to stress. This could also explain why Goniopholis creates shallower marks than alligators, for example, because they might be more stressed in captivity, whereas Goniopholis was in the wild. The next test we did was a species test on the apices. We can see once again a lot of overlap between the different species. However, one here stands out in yellow, which is Mesostops cataphractus. And if you look at the MANOVA results, you can see that they're much, much lower than all of the others, which means they're the most different. This corroborates what we saw on a macroscopic level. If you look at the three other species, they have quite robust teeth, whereas Mesostops cataphractus has much more slender teeth. So this signal is also kept microscopically. We then did the same size test as earlier, but on apices this time. And once again, we see that small individuals are grouped together. They don't have that much variability, whereas larger individuals have more variability. If we look at earlier's graph on the bite marks, we can see that they're very, very similar. So while we weren't able in this study to find a code that would put all of our data together to create direct correlations, we can see that if we succeeded, we would probably have very promising results. It's a future project we have. The final test we did is a wear test. So on this side, we have very worn teeth, and on this side, we have very little worn teeth or with no wear at all. So we can see that the groups are perfectly together. So wear is a very strong signal. What's particularly interesting here is we used all 4 profiles for this test, which means that no matter the direction of the tooth, it still keeps the same signal. So it means that the wear basically removes all the other signals, which makes sense. So in conclusion, apex micromorphology can teach us a lot, not only about the marks that can be produced, but also about the individuals. For example, it can teach us whether or not they had a lot of heterodoncy or not with different morphotypes, their size, their age, tooth wear. They can also teach us, for example, there are other things we'd like to investigate more, like competition between individuals. Does that increase aggression? Things like that. Can we determine if they're scavenging or hunting, for example? Those are things we'd like to explore further. So we can see that this is really interesting for archaeological and paleontological contexts, because we don't always have a lot of information on the bite mark producers. So this is a new method to give us more information. Thank you all so much for listening, and I'd really like to thank the team that helped me with this. I had a lot of help, and also all my co-authors who were mentioned at the beginning. And thank you all for listening, and thank you for VCWAP for letting me speak today. Thank you.